In this video, we will discuss about the tumor-associated macrophages effect on nociceptive neuron function. So in this figure, we will discuss about two kinds of effect of this tumor-associated macrophages on the tumors. First, based on the mechanical sensitivity, whether this TAM affect the mechanical sensitivity on the tumors or not. In the second, we will talk about this nociceptive neuron function. That is, if this around this tumor-associated macrophages, how this nociceptive neuron works, whether they are more work in the sensory perception of pain or they more work towards this neurogenesis. We will understand this panel C first, but first we will start with this panel A. So in the panel A, what it is, they use this IgG2A and CSF1RB antibody. The CSF1RB antibody prevent the interactions of tumor-associated macrophages in the these tumors. So when they check it for the controls, they do not see any significant difference, as the normal tissues does not attract macrophages, but the cancerous cells attract macrophages, and they did the testing for 3 different cancerous cells: head and neck squamous cell carcinoma, melanoma, and fibrosarcoma. And in all the 3 cases, when they use CSF1R, they are able to see reductions in the 50% head movement of the mice, compared to the case where they don't use CSF1R AB, or they use the controlled IgG2A antibodies. That does not affect the tumor-associated macrophages interactions with the tumors. And they did the similar studies with different strategy. In this strategy, they use itgam-DTR-EGFP+DT and the wild-type DT. So here the ITGM is targeted by this DT, that it will degrade the macrophages around the tumors, and in the wild-type DT it does not affect the macrophages, as it is not specific to any kind of cells. So for the control, they do not see any difference, as they do not have any this healthy tissues does not have any attractions towards tumor-associated macrophages. But for the case of cancer cells, they are able to see a clear difference, that is for melanoma and fibrosarcoma, that in the case of ITGM-DTR, they are able to see reductions in the 50% head movement of the mice, or the mechanical sensitivity decreases in the cancer conditions when they reduce these macrophages around the tumors. But this is not the case for wild-type DT or the control. So this indicates that TAM, that is tumor-associated macrophages, affect the mechanical sensitivity of the around the tumors. Afterwards, what they did is they wanted to check whether this TAM also affect the role of these nociceptive neurons or not. So here what they did is they take the single cell RNA-seq analysis and spatial transcriptomic analysis, and they checked, and the question that they asked whether the TAM affect more around the neurons in terms of the sensory perception of pain or the neurogenesis, and they checked it on the cancer cells and the different kind of immune cells. So this is in the center, they keep a TAM spot, and they compare the gene expressions on this TAM at different point of time. So what they are able to see in the cancer cells, the gene expression is more closely aligned with the neurogenesis compared to the sensory perception, except for this 250 micrometer, where it is aligned to both neurogenesis and sensory perceptions. And the comparison is done based on the gene expression in the tumor-associated macrophages. However, for the other conditions or the immune cells, it is CD4 plus T cells, CD8 plus T cells, B cells, dendritic cells, neutrophil and fibroblast, they do not see significant changes at different distance. They checked it for like 50 micrometer, 100 micrometer, 150 micrometer, 200 micrometer, and 250 micrometer. So this could indicate that only in the cancer cells they are able to see a change in the nociceptive neurons gene expressions related to the nociceptive neuron, or gene expressions related to the neuron functions. They are more prone towards this, more close towards this neurogenesis gene expressions compared to the sensory perception of pain or neurons related to the pain. this is what they have find out so far in the part of this figure 2. We will discuss the other part of the figure 2 in our subsequent videos. So, and there we will discuss in detail how this nociceptive neuron functions gets affected by the TAM. So that's all for this video. If you need to understand more about this topic or these results in detail, you can go through the reference that has been provided in the description. Thank you for your time.